We're going to continue our conversation about the immune system, the adaptive immunity. And I mentioned a few times here that, this is antibody, this is antibody, this is antibody, right? We never said what the antibody is. Antibody is a protein molecule, it's a protein that can recognize the antigen. Basically like lock and key idea. Does that make sense? That statement. So this is the typical this is a schematic representation of antibody right here. And this is the actual structure. Okay? So you can see that it has roughly a Y shape, the molecule. Each antibody molecule consists of two heavy, so this is heavy chain, and two light chains. If you would look at the molecule of antibody as a whole, you will see that the molecule has three distinctive fragments. Two FAB fragments that will be here, and one FC fragment. Alright? FAB. AB stands for antigen binding fragments. FC stands for constant fragment. So as you can guess from the name, two FAB fragments can bind the antigen, right? And it turns out that FC fragment can bind to different cells of the immune system. Okay? Does that make sense? Now, what I described to you now is called antibody. It belongs to the class of molecules that we call gamma globulins or immunoglobulins. But structure-wise, antibodies, receptors on the B cell, receptors on the T cell, they all are fairly similar. Does that make sense? So if you take a T cell receptor, it's going to have two heavy chains, two light chains, two FAB fragments, all the same stuff. Am I clear? Now let us see what's going on when cells recognize the antigen. When cells mature, B cells in the bone marrow, right? T cells mature in the thymus. They start to produce receptors on the surface. On the surface of B cell, the receptor is called immunoglobulin D. On the surface of T cells, it is TCR, T cell receptor. The feature of the receptor is that each individual receptor, each type of receptor, let's put it this way, each receptor recognizes a unique antigen. Think about it this way. Each receptor is the unique key that fits a unique lock. And you have billions of keys that will open any lock possible. So far, we cool? So a given cell will have only one type of receptors. So if you have if you take a specific B cell, it's gonna all immunoglobulin D receptors on its surface are gonna be the same. If you take an individual T cell, all T cell receptors are gonna be the same. Does that make sense? Now, if we have 100 cells that are identical, 100 say B cells that are identical and have the same receptor on the surface, this is called a clonal population. Okay, so if you would look at the right bottom side, this cell will represent a particular clonal population. This is going to be another clonal population. Do I make sense? Okay? So each of these bottom cells is going to be clonal populations. Does it make sense? When cells mature, whether they mature in the thymus or in the bone marrow, there is a step called negative selection. During negative selection, now think about this. If you accidentally have an immune cell that will recognize your own proteins as the antigens, that's bad. Does that make sense? This is really bad. No, seriously. Okay? So these cells would be self-reactive. Such self-reactive cells, shown the bottom like this and this, they are eliminated in the bone marrow or thymus. Does that make sense? There's a mechanism of it. So in ideal case, all self-reactive cells are gone. If they aren't gone, what type of disease does patient develop? Autoimmune. Does that make sense? So we got rid of all possible self-reactive cells, and we ended up with a number of clonal populations. At some point, one of these clonal populations will interact with the antigen that matches. Let's say third from the left will interact with this antigen. Does that make sense? Now let's say, so this is the only population that will interact with whatever antigen we're talking about. This moment when it interacts with the antigen is called clonal selection. You select the clone. Why do we call this population of cells a clone? They're all identical. Does that make sense? So what you see on the bottom is just one cell, but imagine you have like 1 million of those cells, and they're all identical, like stormtroopers. Okay? When this interaction happens, these cells start to proliferate. I cross out plasma cells. Okay? Just cross out plasma cells. It's not really correct. Just cross out plasma cells here. Okay? This is called clonal expansion. Do I make sense? So first, there's a clonal selection. Think about this. I'm going to throw some crazy analogy at you. Imagine that you're like a magician, okay? You're in your own castle, and you have a lot of tiny little soldiers on the shelf. And these soldiers fight orcs. This one soldier fights orcs. This one fights dragons. This one fights, I don't know, whatever. Go with you, huh? Whatever you want. Whatever you want. Okay, sorry, I just didn't hear. So, whatever you want. Hobbits, okay? So, say you look out in the window and you see that there are orcs coming. You take this one soldier, and you're a magician, right? And you cast some spells, and these soldiers, you now have not one, but 1 million of all the same soldiers. And they are specifically fitted to fight orcs. So when you pick the soldier from the shelf, that was clonal selection. When you cast the spell and you have 1 million of them, that's clonal expansion. Does that make sense? Good. B cells can do it directly. So B cells can recognize the antigen right away. T cells are picky. They require so-called antigen presentation. We will talk about it. Basically, like if this is an antigen, okay, this is an antigen, empty cell, nothing. This antigen has to be presented to me by so-called antigen presenting cell. Then I will recognize it. That makes sense? Okay. So far, are we good? The most important question. How many antigens are there? How many antigens? 5, 10, 1,000, million, billion? We know about 10,000 different species of bacteria. Only bacteria. And each bacteria has, I don't know how many proteins, and everybody here has slightly different, immune antigenic status. So your tissues are going to be foreign to my tissues, and so on and so forth. And then if you will try to install a cat liver, well, not cat, bad idea. Like a horse liver is probably not a good idea. Give me some animal that is comparable in size to human, like seal, okay? Liver from the seal into humans, it's probably not going to work that well, right? Because of all the antigenic difference. There are probably billions. So you got to have billion, roughly, say billion, different proteins, different antibodies that will recognize billion unique antigens. And each antibody is a protein, and each protein is encoded by a gene. So billion genes in the human genome. We have 30,000 genes in the human genome. So it's definitely not how it works. How would you do that? Mechanism, in my opinion, is one of the most elegant mechanisms that I've seen ever. It's a marvel. Now, let me go back here and point your attention to the fact that antigen binding fragments of antibodies are variable, and they kind of sort of separate. This fragment is constant. So what you really need to change, you can leave the constant fragment the same. What you really need to change are these antigen binding fragments. If you would look at these three keys, they all have the same part here. But this part is different. Not terribly different. It's basically a combination of those teeth, in different crevices and stuff like that. Some basic elements. And you do different combinations, and you can create a huge number of keys. Does that make sense? Same happens in the antibodies. Turns out that we have genes for fragments of the antibody. So red are called V segments, green are D segments, and yellow are J segments. So let's say you pick, I don't know, number 4, this segment from the D, no, this one, this segment from J, okay? And you kind of put them together. There is a mechanism for it called recombination, okay? And then on top of that, you have this V segment. So you've got V, D, and J. And if we give them numbers, just so it's going to be easier for us to orient ourselves. So we pick number 2, pick number 5, and we pick number 2, right? So we have a combination of 2, 5, 2. If we would combine different segments, we could get, I don't know, 1, 4, 3. Does that make sense? Every time there is a VDJ recombination, there is a different variable region produced on the antibody. Does it make sense? Now, you don't need to memorize how many V regions, God forbid. I have no idea. I have no clue. Those numbers every time holy cow, we have a lot. When we pass this slide, I will forget them right away. There's no point in remembering them. The point is, we have a lot of those. Okay? And we have some on the light chain, some on the heavy chain. Okay? So we can combine more than 3 actually. There are going to be combinations of like 5 or 6 fragments. Do I make sense? Turns out that the number of combinations is 300 billion. Which is more than enough to cover possible exposure to antigens throughout our lives. That makes sense? And there's not a lot of genes there. So it takes up just a small, very small chunk of the human genome. I find it incredibly elegant. So please, understand how it works. It's a combination of different genetic fragments that produce eventually a part of the variable region of antibody. We good? Okay. Now, you have a patient. Patient has a disease. You suspect that it can be something dealing with its immune system. You need to know which antibodies to look for. There are 5 classes. In globulin D, the most boring class is the B cell receptor. Okay? I'm highlighting the functions that you need to know for the exam. Am I clear? Like, if you get a question, you're going to get a question like this. Which of the following antibodies serves as the B cell receptor? IgD. Clear? IgE. Allergy. IgE is produced in response to exposure to some sort of an allergen. Will bind to basophils, will lead to the degranulation. They will release histamine, and you have allergic reaction. Ig. So, and this is the point really. When you look, if you want to look deeper, like you have a patient with suspected allergic reaction to whatever, seriously, whatever. So you can isolate specifically IgE from the patient. And you can see if if the patient has IgE, and if these IgE molecules can specifically bind to the suspected allergen. Does that make sense? Nobody does it, ever, because my skin test is much easier. But technically, you can do it. IgA, secretory immunoglobulin found in secretions. It is all over the mucosal membranes. It's basically your watchman. The first antibody that encounters the infection. It exists as the dimer, and it is responsible for the immunity we get from the mother's milk. Now, sometimes I get questions from students that it's counterintuitive. We eat proteins, and proteins get digested in the intestine, right? That's the trick. Structure of the IgA is resistant to digestive enzymes, to the acid, and to the bile. So it remains unaffected. So when we consume milk that contains IgA, we essentially populate the intestinal tract, and we also get it in the blood, and it works just fine, and it provides infants with that passive immunity as long as mother feeds. Seriously. So another argument for breastfeeding, you keep kid protected. IgM, the gigantic pentameric antibody, which means 5 molecules. You can see this. 1, 2, 3, 4, 5 molecules. IgM are the antibodies of the primary response. That's really important. Let me explain why. You have a patient, and I know that sometimes you can correct the doctor if the physician that you work with doesn't know what he or she is talking about. Bring it up. It's really a convenient diagnostic tool. You have a patient, and you suspect that the patient has an infection A. Right now, patient has a flaming disease, and you think that it's a bug A. You draw blood, and you can specifically, those kits are commercially available, you can analyze for IgM. If you find high levels of immunoglobulin M against the bug A, you got your problem. Does that make sense? Because it's the first responder. Okay? If you have seen no IgMs, then infection is caused by something else. Does that make sense? I will draw it in a second when I will tell you about IgG. Because IgG, immunoglobulin G, is the most common class of antibodies. They represent about 70% of all antibodies. They do most of the job, let's put it this way. They're protective, can be found anywhere in the blood, and they are transplacental antibodies that can be carried out from the placenta to the fetus. So they are another way for the mom to protect the fetus. Does that make sense? Now, diagnostic thing. I'm going to draw a really, really tiny graph because I don't have a lot of space. Can you see it on the right bottom? Okay, so this is the antibody levels. This is time. This is the infection point. That's infection. I'm going to draw IgM in blue. So the first thing that you see is the increased levels of IgM, and then they decline. Does that make sense? And next, with IgG, you're going to see this. IgGs are going to go up much later. So if you have a patient in whom you suspect whatever infection with the bug A, the patient comes in and says, I'm kind of really, really sick, and I got sick like a couple of days ago. You can draw blood, check both IgM and IgG levels. What you're going to find in the first couple of days? Probably this. You draw blood 2 weeks later. Are you going to see any IgMs? Probably not. IgGs? Certainly. These 2 blood draws is practically a clinical diagnosis of an infection. It's a gold standard. It depends, you know, it depends on the infection. Sometimes you understand that we don't clinically diagnose all the infections. If you will try to find the statistics on the clinically confirmed cases of flu in states, it's terribly hard. What you're going to find instead is the flu-like illness. It's an official term, influenza-like illness, ILI. Because when somebody during the season, like we have now, comes in and has all the respiratory symptoms, plus myalgia and arthralgia, that's pretty indicative of the influenza. Maybe they will run the test, maybe they will not. It all depends. Does that make sense? So, antibodies. We got antibodies through, let's talk about B-cell response. These are your B cells. And this is your antigen. So you can see that only B cells with the dark blue colored receptors, this, can bind and recognize the antigen. This is clonal selection. Good? When clonal selection happened, clonal expansion starts. B cells start to proliferate and differentiate. So they become more numerous and they also specialize. B cells differentiate into plasma cells and memory B cells. Okay? Plasma cells produce antibodies. We're good? Memory B cells retain the immunological memory about the infection that you encounter. We're good. What you see here is called a primary response. This is what happens when your body encounters the pathogen for the first time. During the secondary response, you already have memory B cells. So in this case, clonal expansion will happen much faster and the levels of antibodies will be much, much higher. This is the generic graph that shows you this. Okay? So this is your primary response, right? And then this is the second encounter of the pathogen, of the same pathogen that was here. Sorry, that was here. You see how higher the levels of antibodies are. That's secondary. Clinical connection, booster immunizations. When you go and you get your first vaccine, produces a certain level of antibodies, and then they start to go down, and then you get second dose of vaccine. The levels go up. Okay? And more memory cells are formed. And then you get, say, third booster. Say hepatitis B vaccine, you get 3 immunizations. And you get a good amount of antibodies, and you get a good amount of memory B cells. So if you ever will encounter hepatitis B virus, you already have a certain level of antibodies in the blood, and you have memory B cells that will respond to the infection incredibly fast and efficient. Does that make sense? Are we clear? Good? Now, what humoral immunity Immunity mediated by B cells is called humoral. There's nothing funny about it. Humor means fluid, liquid. Humoral immunity means that antibodies are soluble. They are in the plasma. Does that make sense? So basically, you can, I'll give an example. First of all, what can antibodies do? Rule of thumb, antibodies never kill. Unlike machete, okay, they never kill. They tag the antigens. So what happens? Say antibodies bind to a virus. They stick to it, they surround it, and the virus cannot do anything if it's all surrounded by antibodies. Does that make sense? Just that's it. It cannot infect anything. It's all blocked. Good? They can agglutinate cells, meaning like clumping them. Okay? When you put cells, when you pull them all together and they all form the clump, there's also not much infection that they can do. They are basically immobilized. They can precipitate antigens. Now that's one of my favorites. Anyone got ever bitten by a venomous snake? Good. Seriously, good. But if you ever get bitten by a venomous snake, if you make it to the hospital, they're going to give you antivenin, which is the drug. It's not just a drug. It's produced by horses. What is done is take a venom from a snake. Have you ever seen that? How snakes donate venom? It's pretty awesome. So they take venom and they inject it into a horse. Since horse is huge, I mean, horse is getting sick, yeah, but not dying. And venom is the main component of a venom, is a protein component. So horse starts to produce antibodies against that protein. Those antibodies are in the plasma. You can bleed a horse, well, the horse, okay? You can collect blood, separate formed elements and the plasma, and basically package that plasma as antivenin. Plasma, humor, will contain antibodies, therefore humoral immunity. When you get the bite, you are given antivenin, basically horse plasma. Antibodies bind to the same proteins in the venom and clump them down. So proteins cannot do much damage, and macrophages can easier munch on them because they're bigger. Does that make sense? Okay. Now this binding actually helps macrophages to phagocytose pathogens. This binding is called opsonization as well. Remember we talked about opsonization with complement? When complement proteins surround the pathogen. When antibodies surround the pathogen, this is also opsonization. You can call it opsonization. All that surrounding it's opsonization. And opsonization labels the pathogen for macrophages to destroy it. Does that make sense? So like macrophages, they just wander around and just don't see anything. Then they notice. They don't need sometimes to notice a pathogen. If they see antibodies surrounding something, that's going to be eaten. Does that make sense? Okay? Antibodies can also enhance inflammation. And they can activate complement via classical pathway. You have a bacteria, Bacteria is surrounded by antibodies. Antibodies activate complement. Complement proteins form membrane attack complex, and the pathogen is dead. Does it make sense? Types of humoral immunity. First of all, humoral immunity can be active and passive. So active immunity when you are exposed to a pathogen. How can you be exposed to a pathogen? What should happen? What are your two options to get exposed to a pathogen? How can you be exposed to a pathogen? Huh? Yeah, infection. Yeah. You can be infected, right? Now that's unfortunate. Or you can get a vaccine. That's also active immunity. So your B cells, your B cells actually recognize the pathogen and produce antibodies. So you get your memory B cells, it's all good. Or humoral immunity can be passive. In the passive humoral immunity, only antibodies, no memory cells. Antibodies are transferred, say from mother to child. Milk, transplacental, this is passive immunity. Or that example that I gave you with antivenin, that's passive immunity. Any transfer of immunoglobulins is the passive immunity. Do I make sense? Immunity, and on top of that, we can say if it's natural or artificial. So say when I get infected, that's the natural immunity, correct? Natural active. When I receive the injection of antivenin after I was bitten by a snake, that's artificial because I get injection. Passive, because the only thing that's transferred are antibodies. Does it make sense? I want you to be able to distinguish between these four types and, match the term and the description, basically, and know what antibodies can and cannot do. They cannot kill. Okay, they label. We good? Is that good? Okay. Next topic on our agenda is T cells. And it involves the concept of major histocompatibility complex. So let's define what this is. I'm going to use the short MHC. Let's figure out, well, major is like a big one, right? What is histocompatibility? Histo. The word histo. Histology, histo. No? Not cell, level up. Tissue. Histo means tissue. Well, compatibility is like compatibility. So tissue compatibility. That makes sense? This MHC is the group of proteins that determine foreign or self for T cells. Because as I mentioned before, T cells cannot simply recognize the pathogen. They need to be presented with a pathogen. And the function of this presentation belongs to antigen presenting cell. Does that make sense? So this is antigen presenting cell. Am I clear? antigen-presenting cell shown on the right here. On the right. This will be infected cell. We'll talk about the picture on the left. Don't pay attention to the left one. This one. Antigen-presenting cell, like dendritic cell or macrophage, eats up the pathogen. Does that make sense? And then, so I'm going to be antigen-presenting cell, right? And this is going to be a pathogen. So I'm eating the pathogen, and then I break it in pieces. And then I present it on my surface as the complex with MHC, major histocompatibility complex 1 and 2. So I have both molecules. Okay. And then T cells will recognize these antigens. Okay. Special cells called cytotoxic T cells will recognize antigen bound to MHC class 1. Helper cells will recognize the antigen bound to MHC class 2. The feature of MHC class 1 is that this complex, MHC class 1, can be found on every nucleated cell in the human body, from hepatocytes to white blood cells to cells of the connective tissue to the epithelial cells, any cell. Does that make sense? Now imagine that I'm gonna need some help here. That I am antigen-presenting cell, okay, and hope is going to be cytotoxic A cell. Okay, so I present to you this antigen, and I tell you this. Basically, what I do here, I tell cytotoxic T cell, this is what you should look for in the body. If the cell, any nucleated cell in the body, carries the same antigen on its surface, it's bad. Got it? No, seriously, I'm kind of teaching. Okay, got it? Now I have two nucleated cells. 1 and 2. Turn around. Can you please present the antigens? You can kill. Which one would you kill?
The red one.
Exactly. Does that make sense? Because you now know I presented you with this. And every cell, every nucleated cell in the human body has MHC class 1. So cytotoxic T cell can, quote unquote, approach any nucleated cell and kill it. If it finds a virus infected cell, it's going to kill it. If it finds a cancer cell, it's going to kill it. As long as it was presented with the proper antigen. Does that make sense? Now, one more. Still, you looking, trying to find what's going on. Blue is the antigen 2. It's not cell. This is blackest cell. Who are you going to kill?
Still the red one?
Absolutely. Because you trained to kill only the red one. If I would train, say, Eric to kill the blue one, then he would represent totally different population of T cells that would kill only blue ones. Does that make sense? So each T cell population, thank you very much, each T cell population can kill only cells that have, that specific antigen on the surface. Does that make sense? So basically, if antigen-presenting cell showed T cells flu antigen, T cells will kill only flu-infected cells. They will not touch anything that deals with measles, that is infected with measles, for instance. Are we clear about it? Okay. Class 2, present only antigen-presenting cells. Only. 3 types. Dendritic cells, macrophages, and B cells. Those are 3 antigen-presenting cell classes that you must know. Dendritic cells, macrophages, and B cells. Clinical relevance. All human cells, as Alexis showed us, all human cells, they display on their surface their own antigens. So basically what that means is, so this is MHC class 1, this is the antigen, and the cell says, I'm fine, I'm self, and I'm not infected. Does that make sense? You do the solid organ transplant. And what happens is, you have organ which cells do this. Are they infected? No. Are they functional? Absolutely. The only problem is that your immune system recognizes them as not self. They're blue, they're not black. Does that make sense? Hence, your immune system will attack and may, and actually will, destroy the transplant. Does that make sense? Good? So, how it all plays out with the T cell response. From the bone marrow, T cells migrate into the thymus. In the thymus, they are selected for not being self-reactive. Are we clear about it? No self-reactivity. And they are separated into 2 groups. CD4 and CD8. CD4 cells can recognize only MHC class 1. Now, CD4 cells, apologize. CD4 cells can recognize only MHC class 2. CD8 only MHC class 1. This ability is called MHC restriction. Does that make sense? Little rule, how you memorize it. I call the rule, it's always 8. 2 by 4 is 8. 1 by 8 is 8. Does that make sense? It's a stupid mnemonic rule, and usually stupid mnemonic rules are remembered the best. Okay? So what happens next? When they recognize the antigen on the antigen-presenting cells, they get activated. So CD4 cells become helper cells. What do they do? Based on the name, they help. CD8 cells will become cytotoxic or killer cells. And they kill. That clear? Okay. Of course, both populations, they will form a set of memory cells. So you're going to have memory CD8, memory CD4. It's all going to happen. So far, do you get the progression? Now, what they do. let's talk about CD8 because they pretty like straightforward. CD8 cell recognizes the antigen bound to MHC class 1 on the antigen presenting cell. This is APC. APC. Okay. Gets activated, gets in the blood, circulates around, finds the infected cell, okay, that has the same antigen bound to MHC class 1. Remember, MHC class 1 is on every cell in the human body. So if it finds infected cell, it's going to kill it. Does that make sense? CD4 cells are more sophisticated. They can recognize antigen that is bound only to MHC class 2. And MHC class 2 can be found only on antigen presenting cells, which are dendritic cell, B cell, or macrophage. So after CD4 cell gets activated, okay, it can recognize antigen bound to the B cell and stimulate its differentiation into plasma cell. Does that make sense? It's like a coach. It's like a coach. B cells, you know, they can become plasma cells and produce antibodies, and it's fine. But CD4 cells, like T helper cells, make this process much better. Does that make sense? We're good. Macrophages. They can phagocytose, but CD4 cells activate macrophages and make this phagocytosis more efficient and guided. Does it make sense? On top of that, CD4 cells here, they produce chemicals called cytokines that will stimulate activation of CD8 cells. Let me ask you a little question. Imagine for a minute that you get rid of all CD4 cells. So which specific population of effector cells you're losing? CD4 means CD4, there's no binding to class 2 MHC, so what you're losing in the bottom? Helper cells. You don't have helper cells, what happens to the B cell activation? No helper cells? B cell, it sucks. It goes down. What happens to macrophage activation? It sucks. What happens to the CD8 activation? It sucks. Okay? You get rid of simple helper cells and basically the entire adaptive immunity. Just tons. Okay? That makes sense? You should ask, how can we get rid of CD4 cells? We heard about HIV. That's what happens. CD4 cells are gone. So just CD4 cells are gone, and adaptive immunity is basically gone. Does that make sense? That's pretty remarkable. Okay? Another important function of CD4 cells is to produce so-called T regulatory cells. They're shown here. So Tregs, as they are referred to sometimes, they needed to dampen the immune response, to decrease it. Think about this. You basically unleash all hell and fury on the pathogen. And how do you feel when you sick? How do you feel? How do you feel when you have a flu? Horrible, right? You know, it's not flu that does it to you, technically. It's your own immune system. You feel sick because of your immune system. So every time you get horribly sick, with all the pains and headaches and fever, think about that your immune system works really, really well. Maybe, you know, it'll make you feel better. So, but imagine for a second that there is nothing to stop your immune system from attacking your own tissues. It's going to be a disaster, right? Your own tissues are going to be destroyed. So T regulatory cells, they control the immune response and kind of bring it down. Kind of don't let it get out of control. Guess what? If your T regulatory cells are not functional, or you have less of it, what type of disease are you going to have? I heard like a faint voice. If Tregs are gone, there is nothing to dampen the immune response, and your immune system keeps attacking your own tissues. Autoimmune. There are multiple studies of which T cells are present in the lamina propria of the intestines in the inflammatory bowel disease. And in these studies, tissues from people with IBD compared to tissues from people who do not have IBDs. These studies are incredibly hard to do because while colonectomy is somewhat a routine procedure for people with IBD, you usually do not nonchalantly remove colons from healthy people, you know. So you have, if there is some other condition that requires the colon removal and there is a fragment of healthy tissue, then you can look at it. Does that make sense? It's really hard. But these studies show very, very convincingly that the number of T regulatory cells in the lamina propria of people with IBD is much lower than the number of Tregs in the lamina propria of healthy people. In healthy people, in us, if nobody, I hope nobody has this condition, but in a healthy patient, Tregs will keep inflammation under control. Is there going to be some inflammation? Quite likely. You can experience colitis, some sort of an infection, but then it subsides. In people with IBD, Tregs are not there, or they're dysfunctional. They can't keep inflammation under control, hence you have this horrible condition. Does that make sense? Okay, we're going to talk about CD8 cells next time.